Hello, today I'm doing my homework because, 126 questions is a few too many for me to get done in class. I've already got it. I'm already about halfway done, but I can finish it in the 15 minutes I have. Apparently, I have an hour 30. There's a protein that initiates. That would be cyclin. Next question. What is the process of programmed cell death? It's the only one that I don't know the word is called Billy Bob Joe. No darn, it was cyclin dependent keto. I never would have gotten that. Pulled apart during chromatids are pulled apart during anaphase, I think. Cell cycle contains checkpoints during the G1. True! Combination to start the cell cycle. Just going into the cell cycle. I don't know. Oh wow, that was correct. I was not expecting that to be correct. The cells in the tissue between the fingers and the toes shows the appropriate type. Stem cells promoting tissue growth. That one, that one, that one. Apoptosis. Condition called cancer. Not grandpa. Which enzyme binds to cyclin during the interphase in mitosis initiating activity? I wonder which one bonds to cyclin. What is the uncontrolled? That one is cancer. That causes cancer. That tries and stop. Those two try and stop it. During which stage of mitosis are the chromatids pulled apart? I think that's anaphase. There we go. Look, I'm on a roll. Known to cause cancer is a carcinogen. One second. Gotta. The answer is carcinogen if it's the one that causes cancer. Spindle checkpoints in mitosis can detect failure of spindle fibers. Monitors for DNA damage. There are no checkpoints during that one is false. I've gotten this question before. Water is not a carcinogen. I mean, technically, everyone who's gotten cancer has drinking water. You're much more likely to get cancer if you drink water. If you think about it, you're much more likely to develop cancer if you drink water. I'm just saying. Like, people that don't drink cancer don't no people that don't drink water don't tend to get cancer. like occurs in cells that are damaged beyond repair, including cells with DNA damage that could lead to cancer. Oh, that is apoptosis. And pop goes the A cell, and apoptosis goes the A. Pop goes the ptosis. Has the same risk. More who inherits one or more genes is at a higher risk of developing cancer than someone. I think it's that one. Yeah. When cells do not respond to the normal cell cycle control mechanisms, a condition called cancer can result. The DNA on chromosomes is arranged into segments called genes. I have to move this bag of pretzels. It's in my way. I have to move this other bag of pretzels. Don't ask me why I have 2 bags of pretzels. It's the uncontrolled growth and division of cells. Cancer. In humans, each parent contributes 23 chromosomes to a cell. This results in 46. It's obviously 69. My science teacher has a sense of humor with that one. A substance or agent known to cause cancer. Carcinogen. Actually, the correct answer is aspartate. I'm joking. That one's not actually more likely to cause cancer than most things. The chromosomes that make up a pair, when chromosomes are called sister, I think is the correct word. No, homologous. I'm thinking of a different kind of chromosome then. Oh no. Although, I wonder, literally the same question twice. I hate this because it gives you the same questions twice, meaning you have to complete actually twice the amount that it says up here. Because each of these it gives you twice. So that's actually 126. They carry genes. Individual here, one or more is at a higher which is high 6 a gamete. It describes the sequence of DNA on chromosomes that control the production of proteins. Genes. Cell with half number of chromosomes is called a haploid cell. Human body cells have 49 chromosomes, yes. In which process does one haploid gamete combine with another haploid gamete? Fertilization, yes. Ayo. How does the fertilization happen? We don't talk about that at school. What term describes the chromosomes that make up a pair? One chromosome from each parent. Homologous, yes. Select statements about the homologous chromosomes shown here that are all true. Okay, so they gotta be true. They gotta be true statements about these chromosomes. The genes that code for earlobe type on each chromosome might not code for the exact same type. The gene for earlobe type will be located on the same position. Genes that code for earlobe type on each chromosome will code for the exact same location. Carry genes for any given trait at the same location. Correct. It's called a diploid. Gamete. Frick, you can't just smash enter. Paired chromosomes that determine an individual's gender are... That's the wrong word there. That should be sex. Cells. What determines gender is personality. Hashtag trans rights. What? Oh. What is a cell with n number of chromosomes called? Haploid. Is a female. The process by which one haploid gamete combines with another haploid gamete is called fertilization. Protective cap at the end of a chromosome is called it's called like homologous. What type of cell contains two diploid telomeres? Might be involved in both aging and cancer. Telomeres protective function. Genotype. What is the protective cap found at the end of the eye? It's the telomere, obviously. It's called bad. Siamese twinning. Telomeres are the site of protein synthesis in a cell is not true. Can result in extra copies of certain chromosome. Mistakes. Darn, it was non-disjunction. I hate this. Paired chromosomes are sex chromosomes. Non-disjunction of autosomes. Karyotype. It's caused by that did not answer the question. Oh darn, it was Turner's. It's called a telomere. Describes cell division during which sister chromatids fail to non-disjunction. occurs only. If not, the resulting gametes will not have the correct number of chromosomes. Extra copies of certain chromosome. Autosomes. I saw that word elsewhere, and I think that's the correct word. Yeah. Semi-conservative replication, unwinding, base pairing, and joining. Yes. Let's go. Disjunction of sex chromosomes. Turners. Kinfelters. Haha. During DNA. I wonder what the answer to that. God. I was technically correct. DNA replication is semi-conservative. In fact, semi-conservative is probably less specific than DNA, meaning that I probably got that right. Cell division during which sister chromatids fail to separate properly is called non-disjunction. Oh my god. Single-stranded DNA bias, like DNA unwinding the single-strand binding protein to single-strand binding proteins associated with the DNA strands together during replication. Helicases are unwinding and unzipping the double helix. Let's unzip the hydrogen. I believe just the helix unwind after RNA. And they never adds 4 of them. Can result in extra copies of it. My god. Non-disjunction. Whoa. I'm probably going to have to copy that. And then catalyzes the addition of appropriate nucleotides into the new DNA strand. That is primase. Wait, it's catalyzes. Yeah, I'm just going to say primase. Oh, it was polymerase. Darn. Process of unwinding. Non-disjunction. Failure. Low. Darn. It was base and pairing. Non-disjunction of sex. That's Turner's. Lagging. High. Next question. During which process do parental strands of DNA separate, serve as templates, and produce DNA molecules that have one strand of parental DNA and one strand of new DNA? Semi-conservative. It's called an RNA primer. I wonder what it is. It's not sure of itself. Polymerase? Oh darn, it was ligase, the enzyme. DNA polymerase. My god. Process of conservative replication occurs in 3 main stages. Why does it give me 2 of each question that just makes it take twice as long? Prokaryotes. Circular DNA strand is opened at 1 point of replication. I don't know. We didn't talk about this. Just gonna say that. No. Which statements about the lagging strand of DNA true? The Okazaki fragments. The lagging strand is synthesized discontinuity. Okay. 3' to 5' direction. The lagging strand is built. Okay, let's go. I've got 4 more topics. What is a short strand of DNA synthesized discontinuously in small segments in the 3' to 5' direction by DNA polymerase? Okazaki fragment. That is a oil company right there. Ligase. Oh, okay. That was not intentional. Hold up. Which enzyme catalyzes the addition of appropriate nucleotides to the new DNA strand? That was polymerase. I'm not even learning the material, I'm learning the questions. It's like the statements about eukaryotic DNA replication that are true. Unwinds in multiple areas. DNA ligase? I swear if it makes me get this wrong because of the question mark, yeah. DNA. I don't know. Anyway, guys, that was my homework.